The first step of using your new P51 Molecular Fluorescence Viewer is assembly. Each P51 comes flat packaged in a cardboard box, and assembly is fast, easy, and fun. After opening the box, make sure you have a circuit board, an LED strip, a spacer, an insert, a viewer, and one yellow and one orange filter. To power your P51, you can use either a USB-C power supply or a 9-volt battery. If using a 9-volt battery, you'll want to keep it handy during assembly. First, start with the viewer. Fold flap 1 down into the viewer. Fold side flaps 2 down and interlock with flap 1. Fold flap 3 down. Flap 3 will interlock under flap 1. You may need to push hard. The viewer base should now look like this. Turn the viewer over and fold down the cover flaps. Finally, close the viewer. The attached Velcro should hold the lid closed. Next, continue with the P51 insert. Peel off the protective liner on the back of the LED strip to expose the adhesive. Making sure that the LED strip wires are in the correct orientation as marked on the insert, stick the LED strip where indicated. Next, place the circuit board by peeling back the protective liner from the Velcro. Do not remove the Velcro from the insert. Place the circuit board so that the white lines on the circuit board align at the top and on the side of the insert. The USB port and switch will overhang the insert slightly. Next, flip the insert over and fold up at creases F2 and F1. Insert tab A into slot A. Fold the insert across F3 and F4 about 90 degrees each. Fold the side flaps and engage them at point B and interlock. Repeat on the opposite side with point C, making sure that the cables come up and out behind the flaps of the insert towards the circuit board. Insert the LED cable into the mating connector on the circuit board. If you'd like to use your viewer with a 9-volt battery, you can now snap that battery into the circuit board. If you're going to power your viewer with the USB-C cable, no battery is needed. Finally, peel the protective film back from both sides of the yellow and orange filters. These filters are interchangeable depending on what you're looking at in your P51 viewer and what the lab protocol specifies. Insert the filter against the inside of the window. While holding the filter in place, install the insert in the viewer with the window facing the front opening and the holes of the insert facing up. The fit will be tight, so some pressure is needed. You may want to tilt the insert sideways when inserting into the viewer so that the USB port and power switch enter the viewer first. Place the spacer behind the insert to secure it in place. The spacer will fit loosely inside the viewer. Finally, make sure you can reach the power switch and the USB port from the side opening. If you can't, remove the insert and realign the circuit board. If using a USB-C cord instead of a battery, you may now connect the cable to power your P51 viewer. Now your P51 Molecular Fluorescence Viewer is ready to go. You can use it with a number of fluorescence-based labs from miniPCR Bio, including our BioBits Central Dogma Lab and our Beta-Gal Glow Enzyme Reaction Rate Lab. To learn more about the cool science concepts you can explore with your P51 viewer, you can visit minipcr.com.